All right, we're going to start thinking about this question that involves the plasmid PBR40. So the question asks us to consider this plasmid, which has been transformed into E. coli cells with or without restriction digest. And we need to answer, will the E. coli cells be able to live and grow in each of the following conditions? Let's first take a look at the plasmid in order to answer this question. All right, so PBR40, the name of the plasmid, and many plasmid names begin with a P for plasmid. And what's in this plasmid here? We've got an origin of replication to start, and that's a good thing to have because it means that a bacterial cell which picks up this plasmid will be able to replicate it, make many copies, and pass it on to the daughter cells. Terrific. In addition to the origin of replication, in yellow here, we've got an antibiotic resistance selectable marker. And in this case, it provides us resistance to the antibiotic tetracycline. And then in blue, we have a second antibiotic resistance marker for the antibiotic kanamycin. So without these resistance genes, E. coli cells would die in the presence of the antibiotic kanamycin or in the presence of the antibiotic tetracycline. But thanks to these genes, they'll be able to survive and grow. Now, in addition to those two genes, note in the kanamycin resistance gene, we have two additional features. We have a BAMH1 cut site and an XHO1 cut site. And these are restriction enzyme cut sites. Terrific. So now that we know what's in the plasmid, let's consider what happens to our E. coli cells when we are transforming or introducing this plasmid into the cells. So first of all, let's look on the left here on the top line. When we have no DNA that's been added into our E. coli cells, in the presence of nutrient-rich media on regular old bacterial plates, we'll have growth of our E. coli cells. But once we add in the antibiotic tetracycline, we see no growth of the E. coli. Because remember, these cells have had no plasmid transformed into them. No growth in the presence of tetracycline, no growth in the presence of kanamycin, an extra double no growth in the presence of both tetracycline and kanamycin. Now, when we look at the cells that have been transformed with intact PBR40 plasmid, we can see that they grow as expected in the nutrient-rich media. But when we add tetracycline to the media, now these cells are able to grow because they have a resistance gene that will help the E. coli cells deal with the presence of tetracycline. Similarly, in the presence of kanamycin, our antibiotic resistance gene found in PBR40 will mean the cells can grow. And they can even grow in the presence of both antibiotics together, since we have separate genes to help us deal with these two separate antibiotics. All right, now let's think about what would happen if we cut PBR40 with BAMH1 and XHO1 and then added it to E. coli. You can pause the video for a moment here if you want to think about your answer. So you can see here that in the presence of regular old media, no antibiotics, these cells would be able to grow without issue. But in the presence of any of the antibiotics, tetracycline, kanamycin, or both tetracycline and kanamycin, we get no growth of the E. coli cells. The reason this happens is because by cutting with BAMH1 and XHO1, we're opening up the plasmid not to be circular, but to be linear. And bacterial cells don't know what to do with linear DNA. It's likely degraded and broken down. So even though it's only the kanamycin gene that's been cut by these restriction enzymes, the entire plasmid is no longer functional. So the cells still don't have any tetracycline resistance. All right, then let's think about what would happen if the cells were cut with BAMH1 and then treated with DNA ligase before PBR40 was added into the E. coli cells. You can pause the video now if you want to think about what would happen in this case. In this case, we'd have growth of the cells under the no antibiotic conditions as expected. but we're going to have growth in the presence of the antibiotics as well. The presence of tetracycline, yep. The presence of kanamycin, absolutely. And the presence of both of those antibiotics together also results in growth from these cells. And that's because by treating this plasmid with DNA ligase, we're able to restore the phosphodiester backbone that was cut by the restriction enzyme BAMH1. And it's the same as inserting an intact copy of PBR40. We restored the original function of this plasmid. All right, here's one more challenge to think about. What if the plasmid looked like this, and rather than a BAMH1 site and an XHO1 site, we had two BAM cut sites in the kanamycin gene? Now I'm asking you, if we treated PBR40 with the BAMH1 restriction enzyme, then ligated it back together and added it to E. coli, would we see growth? Well, in the presence of nutrient-rich media, absolutely we'd see growth. But once we start adding in antibiotics, the story changes. So in this case, our cells would be able to grow in the presence of tetracycline, but not grow if there was any kanamycin present in the media. So in this final condition here, even though the cells would be able to live in the presence of tetracycline, because there's additionally kanamycin here, they would still die. And note that the reason that would occur is because BAMH1 is cutting at two places in this plasmid, which means this little in-between piece is going to be deleted. And it's unlikely when we treat with DNA ligase that this tiny inserted piece would be restored to the plasmid. Instead, a deletion would be created in the kanamycin gene. And so even though the plasmid is returned to be intact, the loss of this region of the gene means that the kanamycin resistance is no longer functional, although the rest of the plasmid is, the origin of replication and the tetracycline resistance gene. So that's why we're able to see growth in tetracycline, but the kanamycin resistance is no longer functional when we have both BAMH1 digestion sites. I hope this helps you think about cutting and pasting of plasmid DNA with restriction enzymes.